Good morning everyone and welcome back to CEC Gurukul lectures. I am Dr. Kumar Shantanu, Associate Professor of Botany from Deshbandhu College, University of Delhi. In this second part of our lecture on Selaginella, we shall continue our discussion on various aspects of biology of Selaginella. Up till now, we have seen the systematic position of Selaginella, the habit and habitat of Selaginella in our previous lecture, and also the morphological aspects of Selaginella species. In this current lecture, we will continue our discussion from the morphology of rhizophores. A rhizophore in dorsiventral species, some of the specialized long leaflet root like structure appear, which are termed as rhizophores. It develops from group of meristematic cells present at the dichotomy of branches and they grow downwards. They are positively geotropic and it develops adventitious root at its terminal end. Rhizophores lack the root cap, so they are not the true roots per se. In the diagram you could see that these leafless branches, they arise from the stem, grow towards gravity, and the terminal positions, they have multiple branches which could be termed as adventitious roots, and from these roots they absorb water and mineral and they also support in anchorage. As far as the roots in Selaginella is concerned, most of the roots are adventitious in nature. Adventitious roots originate from the swollen base of hypocotyls and directly form the stem and also form the tip of rhizophores. They arise endogenously and branch dichotomously. The roots have root caps and root hairs. The roots are having endophytic fungus which are present in its cortical cells. So most of the Selaginella species, they have an endosymbiotic association with fungi. Here in the diagram you could very well see multiple roots coming out and performing the function of anchorage and also absorption of water. Now let us talk about the internal structures of various parts of plant body of Selaginella. Let us first talk about the anatomy of stem. Internally, the stem is differentiated into mainly epidermis, cortex and stele. Epidermis is thick-walled, prosenchymatous in nature and they lack stomata on it. On the outer side of the stem, a thick cuticle is present which helps in protecting the stem from desiccation process and evaporation of water. Just beneath the epidermis lies multiple layers of cells called as cortex. The outer 2 to 4 layers of cortex consist of thick-walled sclerenchymatous tissues called as hypodermis, whereas the rest of the cortex consists of thin-walled green polygonal parenchymatous cells. Here you can see in the diagram that the outermost layer of the stem has one cell thick epidermis and inside this epidermis lies multiple cell layers called as cortex. Now in case of stem, the endodermis is also present In young stem, endodermis is single layered, which surrounds the stem. The stele is those structures which bear xylem and phloem. In case of mature stem, the cells of endodermis elongate in radial directions and separate from each other, and they form broad air spaces. The trabeculae possess Casparian thickenings. The trabeculae are unicellular, but sometimes many become multicellular filamentous structures due to formation of tangential walls. The stem could be monostelic, it could be distelic, or it could be polystelic. Single flattened or ribbon-like stele could also be found. Such type of stele are found in Selaginella chrysocollus. In case of Selaginella selaginoides, the stele is single cylindrical with polyarc conditions. Also, in case of Selaginella selaginoides, in some of the species, we do encounter single cylindrical stele with endark condition of xylems. Here we can understand the anatomy of stem with the help of this diagram. So, the outermost layer of the stem is termed as epidermis. It lacks any stomata, so there is no discontinuity because of the presence of any guard cells. So, they lack any guard cell, and outside the epidermis lies a thick coating of waxy substance called as cuticle. Below the epidermal cell, there are 2 to 4 cell thick sclerenchymatous tissues of cortex. These sclerenchymatous cortex help in providing mechanical strength to the stem. Below the sclerenchymatous cortex lies thin-walled parenchymatous tissues of cortex, and there are innermost layer of endodermis. These endodermal layers, they elongate and get separated from the pericycle, and these elongated cells, they are extensions of endodermal cells. These elongated endodermal cells are called as trabeculae. Because of the elongation of trabeculae, the endodermal layer gets separated from the pericycle and there is a air space created. These air space are a unique feature of stem anatomy in case of Selaginaceae and the genus Selaginella. Now, each of these stems could have one single stele, as it is shown here in this diagram. This stele has a pericycle, the outermost pericycle, and inside this pericycle, in the center, there is xylem tissues. The xylem tissues, they have protoxylem at the periphery or towards the edge and metaxylem in the center. Surrounding these xylem tissues are found phloem tissues. So, in the center there is a xylem surrounded by phloem tissue, surrounded by pericycle layer, and this pericycle is separated from the endodermal tissue, endodermal layer, with the help of trabeculae. The air space between endodermal layer and pericycle is termed as air space. There are certain species of Selaginella which have 2 or more than 2 stele. Such conditions are called as distelic conditions or polystelic conditions. In case of distelic conditions, 2 steles run parallel and joining at the points of dichotomy, such as Selaginella kraussiana. And in many flattened or ribbon-like species, the steels have polystylic conditions, such as Selaginella wildenovi. Here in this diagram, you can very well see two different steels found in one stem. So such stem are called as distelic stem. Here, and there is one steel here, both are found inside the common cortex. Each steel has a central solid core of xylem, which is surrounded by phloem and pericycle. This is what we have seen in the diagram. Phloem consists of parenchyma and sieve cells, but the companion cells are absolutely absent. And as far as the xylem in case of the steel is concerned, the xylem happens to be diarch and exarch. The metaxylem occupies the central position, whereas the protoxylem lies towards the periphery. The metaxylem is composed of scleriform tracheids, whereas protoxylem has annular and spiral tracheids. A few species of Selaginella are remarkable in having true xylem vessels in their xylem, such as Selaginella rupestris, Selaginella oregana. Although secondary growth is absent in Selaginella, but secondary xylem elements have been reported in basal portion of the stem of Selaginella selaginoides. So we can see that there is an beginning of secondary growth happening, and this will gradually evolve into the formation of secondary growth in higher plants. Now let us talk about the leaf anatomy. In case of Selaginella, the leaf is very small, simple and dorsiventral. The leaf consists of prominent upper and lower epidermis. The stomata are mostly present in the lower epidermis. Mesophyll cells consists of thin-walled spongy parenchyma with large intercellular spaces. The cells of epidermis and mesophyll contains variable number of chloroplasts. In a few species, each cell contains single cup-shaped chloroplast, which is associated with numerous spindle-shaped pyrenoid like those which we have seen in Anthoceros also. This can be understood with the help of the diagram. Here in this diagram, we can see that each leaf in its cross section or in its transverse section, it has an upper epidermis. The epidermal cells are more or less barrel shaped in nature, and they are lying horizontally with each other. And also there is a distinct lower epidermis. On the lower epidermis, we mostly encounter stomata, which can be identified with the presence of guard cells. In between the upper and lower epidermis lies mesophyll cells. Mesophyll cells, they are parenchymatous cells which contain multiple chloroplasts. Because of the presence of chloroplasts, they are the major sites of photosynthesis. There are large air spaces which are found between mesophyll cells, and in the center of every leaf, a very prominent vascular bundles can be seen. These are xylem tissues surrounded by phloem tissues. Similarly, let us talk about the anatomy of ligule. As we have already discussed, ligules are found at the base of young leaf and young sporophylls. So Ligule is a small appendage which consists of two parts, the basal, glossopodium, and the upper body. The glossopodium is circular portion embedded in a cup-shaped sheath. The cells of sheath are tubular. The glossopodium has vertically elongated cells. The body of ligule consists of polygonal parenchymatous cells filled with dense protoplasm. Here you can see in the diagram that the basal cup-shaped structure, it has elongated glossopodium, and then parenchymatous cells are found on the upper part of the ligule. As far as anatomy of the rhizophore is concerned, in transverse section it shows circular outline, and it has a thick walled epidermis and the cortex with outer two layers of sclerenchymatous and 2 to 3 inner layers of parenchymatous cells. The endodermis is followed by a single layer of parenchymatous pericycle. The xylem is surrounded by phloem, which consists of sieve cells and parenchyma. The epidermis consists of large thin-walled cells. Few cells of epidermis elongate to form root hairs. The cortex in root mainly consists of thin-walled parenchymatous cells. The sclerenchymatous hypodermis is also present in sometimes. The endodermis is followed by 1 to 3 layered pericycle. The stele is protostelic, that is, central solid core of xylem is surrounded by phloem, and xylem is monarch. That means only one group of protoxylem is found, and the xylem is also exarch in nature. Now this can be seen with the help of this diagram also, where the outermost layer gives rise to root hairs. The cortex has sclerenchyma and parenchyma, followed by pericycle and endodermis, and in the center lies monarch xylem surrounded by phloem. So this was all about the anatomy of Selaginella. Now let us talk about a very important aspect of biology of Selaginella, that is reproduction. As far as reproduction in Selaginella is concerned, it can take place in two various methods. The one is called as vegetative reproduction. Vegetative reproduction takes place in Selaginella by three most common methods. One is called as fragmentation. So what happens in fragmentation is, under humid conditions, the plants develop roots and adventitious buds on prostrate branches. These prostrate branches break their connections from parent plant and grow as separate plants. The second method of vegetative reproduction is with the help of tubers. Tubers are swollen structures that may develop at the apices of aerial, vegetative and underground branches. Tubers store food and survive under adverse conditions and grow into new plants under favorable conditions. The third and most common method of vegetative reproduction is with the help of resting buds. Some species develop very compact resting buds at the apices of aerial vegetative branches. On the onset of winters, they give off rhizophore that bear roots. These resting buds survive under adverse environmental conditions. They grow into new plants on the onset of favorable conditions. Now, as far as the sexual reproduction in case of Selaginella is concerned, sexual reproduction essentially involves spores. The spores are produced in a closed structure called as sporangia. Sporangia on plant body produce haploid spores. Selaginella is heterosporous in nature. That is, it produces two different types of spores. One is called as microspores, the other one is called as megaspores. Microspores germinate to produce male gametophyte and megaspore germinate to produce female gametophyte. The microsporangia bearing leaf, they are called as microsporophyll, whereas megasporangium bearing leaf, they are called as megasporophyll, and they appear on the terminal end of main axis or lateral branches. And these compact megasporophyll and the clusters of microsporophyll, they are termed as strobilus. Sporangia are born singly in the axils of microsporophylls and megasporophylls between the ligule and in the same axis. These sporophylls are present in a compact apical portion of the shoot and they are termed as strobilus. You can very well see in the diagram where you can see the compact leaves or sporophylls which are found at the terminal positions. Here you can see the main axis of Selaginella in dorsal view and in the ventral view. When these leaves bear sporangia, they are termed as sporophylls and they become compactly arranged, and these compactly arranged sporophylls are termed as strobilus. The microspores are produced in large numbers inside microsporangia and megaspores are produced in small numbers inside megasporangia. Sporophylls differ in shape and size from the vegetative leaves. The microsporangium is comparatively small in structure with a short stalk and with spherical body. It has two-layered wall. An outer wall consists of chlorophyllose thick-walled cell layer and the innermost layer is termed as tapetum, which serves nutritive function. The cavity of microsporangium is filled with a large number, somewhere around 600 to 2000 small microspores. In case of megasporangium, the megasporangium is a comparatively large and it has a short stalk and large spherical four-lobed body which has two-layered wall and tapetum. Megaspores are large in size, but their number remains small as compared to microspore. Here you can see the megaspore and microspore inside a megasporangium and a microsporangium. Now both sporangia dehisce by vertical splitting of sporangial jacket into two valves. Spores start germinating in sporangia and liberate in semi-germinated stage. The spores are liberated and scattered by wind. Microspores are thrown in small masses near mother plant and megaspores are thrown to a greater distance. The gametophytic generation begins with the production of haploid spore. Microspore and megaspore germinate to form male gametophyte and female gametophyte respectively. The male gametophyte is represented by microspore. They are tetrahedral and smaller in size. It starts germinating within microsporangia, but the growth of gametophyte occurs after they are shed. The spore divides and develops directly into antheridium, and biflagellated anthrazoids are produced inside antheridia which swim in film of water and reach to archegonium of female gametophyte. Here you can see in the diagram that each microspore is a representative of male gametophyte. They divide and give rise to biflagellated antherozoids. As far as female gametophyte is concerned, they are represented by megaspores. They are tetrahedral with a triradiate ridge and comparatively large in size, that is 1 to 5 mm in size. They germinate within microsporangium and liberated in semi-germinated stage. It develops into a pad of tissue still partly enclosed in spore walls. Archegonia develop on the exposed part of the pad, and each archegonium has a short neck of 2 tiers of 4 cells each and basal broad venter. The upper tier of 4 cells function as cover cells. The axial row consists of single neck canal cell, and venter has a ventral canal cell and egg. The complete archegonium except cover cell is embedded in cellular cushion of female gametophyte. You can see this in the diagram that megaspore tetrad, and each megaspore represents a female gametophyte. They germinate, and archegonia is always embedded inside the endosperm, and after fertilization they form the embryo. After fertilization, the biflagellate antherozoid swim in the film of water, and they are attracted towards some chemical substance secreted by archegonia of nearby female gametophyte. They swim down the canal of open archegonium, but only one fuse with egg, forming diploid zygote. The diploid zygote is the first cell of sporophytic generation. It secretes a protective wall and grows into an embryo. The development of embryo is endosporic. That means the apex of embryo being directed inwards away from the mouth of archegonium. The zygote then grows by various cell divisions and develops into a young sporophytic plant body. So this is how we can summarize the entire life cycle of Selaginella. The sporophyte is there, which has a strobulus. On the strobulus we have male and female, we have microspore and megaspore representing male gametophyte and female gametophyte. They germinate, giving rise to archegonia and antheridia. Antheridia fertilizes the egg inside archegonium. Embryo is formed. The embryo germinates and giving rise to the young sporophyte. So this is how that entire life cycle of in case of Selaginella gets completed. I hope I have been successful in giving you an outline and basic idea about the biology of Selaginella. So thank you so much for joining us.